Hi students, today we are going to see glucose sensor. I am Dr. S. Rekha, Associate Professor, RMD Engineering College. I'm happy to meet you all. Let us see the topic. Let us know what is biosensor first. So this biosensor was first found out by Leland C. Clark, who is the father of biosensor. So what is a biosensor? Any sensor which analyze the changes inside the body, human body, we can call it as biosensor. Any living body, we can call it as biosensor. And that can be converted into electrical sensor, signals, right? Any sensor finally converts into electrical signal. So the biological process can be any biological element or material like enzymes, tissue, microorganisms, cells, acid, etc. Okay, so one example of biosensor is glucometer. So what is this biosensor made up of? So outside the environment we have the sample, and this side outside the environment we have the display screen. And what is biosensor encompasses? It has a bioreceptor and it has a transducer, and we have the signal processing. So bioreceptor receives the signal from the sample, and that is converted by transducer to signal, and this signal is displayed on the screen. So the signal processing, it is electrical signal we get. Glucose biosensor. It is an analytical device which is used for detection of blood sample, that is glucose present in the blood sample. Okay, it is widely carried out by electrochemical methods. So there are three kinds of electrochemical method with which you can detect the glucose in blood. First one is potentiometric biosensor, where we use ion selective electrode and we measure the potential. Okay, next one we have amperometric biosensor, where we measure the electric current associated with the electron flow. And third we have conductometric biosensor. We measure the change in conductance in the medium, and based on that we measure the amount of glucose present in it. Principle behind the glucose sensor is, first one is glucose undergoes oxidation reaction in presence of GOX to form gluconic acid and hydrogen peroxide. So we are going to sense this hydrogen peroxide. This decomposes at the potential of 0.7 volt versus silver chloride reference electrode. So oxygen is formed and 2H plus ion and 2 electron is released. So this, whatever the conversion factor, based on that what is the amount of glucose we can identify. That is, in the first step we have the hydrogen peroxide removed. So what is the quantity of hydrogen peroxide can be measured by the second process. So indirectly we measure the presence of glucose. More the hydrogen peroxide decomposition, more will be the glucose, or more is the oxygen or H plus ion formation, more higher will be the glucose in the blood. So different types of glucose sensor based on their mechanism or of action. So we'll see. So this can be enzymatic glucose sensor or non-enzymatic glucose sensor. Basically research we started with enzymatic glucose sensor. So we have first generation glucose sensor. I said GOX in the previous step, right? That GOX is enzyme. Okay. Second generation glucose sensor, that is after first generation we have done. We have overcome the inabilities in the first generation. So we call it a second generation glucose sensor. And whatever inability we had in second generation, that was overlooked. And we had the third generation glucose sensor. And whatever problem we had in these method, so we wanted to move away from the enzyme. We wanted to remove enzyme itself. So we have a fourth generation where we have removed the enzyme, but yet it measures the glucose in the blood. So these are the types of glucose sensor. All the four generation you can understand better from this equation. This is taken from, this diagram is taken from a research paper which gives you a clarity of all the four generation and the mechanism. let us see the mechanism. So glucose is the input. So glucose is converted into gluconic acid with the help of the enzyme, which is GOX. Okay. So this is the active site. On this active site, we also add something called cofactor, right? So cofactor get reduced. When something is oxidized, something else has to get reduced. So cofactor get reduced. Now this cofactor has to want to come to its original position. This gets oxidized and high oxygen gets reduced to hydrogen peroxide. So oxygen is in the input. Glucose and oxygen is the input. So this oxygen gets reduced to hydrogen peroxide. So this hydrogen peroxide releases the electron. This electron is measured by the sensor, and amount of electron decides the amount of glucose present in the, so this is the first generation. So indirectly we were sensing the glucose. So now there was an hurdle in doing this process. Okay, this we need to apply a zero point 0.7 voltage, which was very higher, and always there was no 100% oxidation of hydrogen peroxide. Sometimes we had insufficient supply of this oxygen. So we were not able to measure it accurately. So then we came up with the second generation where we eliminated this process itself, hydrogen peroxide, because oxygen is limitedly available. So we wanted to eliminate this process. So what we did, we gave glucose as the input, okay, to form gluconic acid and enzyme. With the enzyme and cofactor remained same. Along with the cofactor, we added one more component into it, which is a mediator, which is instead of this step, we remove that step with the mediator. So what happens now? Cofactor gets reduced. Now this cofactor wants to get oxidized. So while it is getting oxidized, mediator which is added gets reduced. So now this mediator must be sensitive to cofactor as well as to the sensor. Okay. So when this mediator is getting oxidized, it releases electron. So this electron is measured by the sensor. Amount of electron released is directly proportional to the amount of glucose present in the blood. Okay. Again, here we have a difficulty that we have added one more component into it. Okay. Initially we had enzymes and cofactor. Now we have also added the mediator. So now again we were not able to get a very accurate answer. So they went with the third generation product, where we removed this both the product cofactor as well as mediator we wanted to remove, and directly we wanted to measure the electron. So what they did, they removed it and they went with the phenomena where they were able to convert gluconic acid and enzyme. This cofactor alone get oxidized. When cofactor is reduced and when it is getting oxidized, release the electron, and that is sensed by the sensor. Okay. So this electron can be sensed. So this is directly, the step is reduced here. Third generation is working good. Mostly available in the commercially viable is second generation. Okay. Now we have worked with third generation. Still we want to be simplified with the technology. What they do, just taking gluconic acid. So I said no, non-enzymatic. Until now we had enzymes, and fourth generation is non-enzymatic. Glucose converting to gluconic acid itself, it can release electron. This electron can be measured using the sensor. So that is fourth generation sensor. This is under development. Fourth generation is under development. If it is getting developed, then it will become the simplest one as well as cost effective one. I hope so you have understood what is how this glucometer measures this glucose in the blood. Whatever I have explained, it is given in the theory form in this page. Okay. Anyhow, you want, I'll read out certain important point. Okay. So here, to function as a catalyst, GOX requires a flavin adenine dinucleotide as redox cofactor. So redox cofactor added is flavin adenine dinucleotide. Okay. FAD functions as an initial electron acceptor and it is reduced to FADH2, right? So that which I have explained, we have explained in the diagram. So I said no, the potential is 0.7 volt is required and limited oxygen supply. Because of this, we are not able to measure it in a proper way, accurate value. Then generation 2, whatever explanation I have given, and the same thing we have written in words. The mediator used is ferrocene, methylene blue, etc. Okay. Then we have glucose detection, how this is done, mechanism. So it is for first generation. So we have glucose molecule, we have oxygen. These two are the input. When it goes to the GOX enzyme and it gets reacted and gluconic acid is formed. And since we have given oxygen also, it forms hydrogen peroxide. This is hitting the electrode where this hydrogen is forming the oxygen and electrons are released, and that is measured here. And third generation, where we remove cofactor as well as mediator. Directly we are trying to measure it with the help of nanoparticles. Okay. So here we are using nanoparticle. Then we have fourth generation, which undergoes generally not even enzymes, we are using direct measurement, right? So this is under research. Sensor consists of several layers. This, whatever in the figure we have explained. The glucose test strip consists of several layers. Let us see what are these layers in detail. So we have the base substrate, which is made up of PET. Okay, and over this PET surface, we have the mediator layer. As I said, the second generation is generally applied to the society, and the third and fourth generation are in research. Okay, yet not yet commercialized. So mediator layer is given a coating over the base substrate. So mediator, for example, ferricyanide is coated over the base substrate. Over that we have enzyme layer, okay, where the oxidation of glucose take place. So glucose oxidase is the example for an enzyme where the reaction take place. Then over that we give a protective membrane again, which is made up of PET. Okay. Then over that we spread a sample spreading layer, that is nylon mesh, over the surface so that the blood which is a sample which is given easily sucked in. So for spreading the blood into it, so for that we apply this layer nylon mesh, and all over that top cover film, and that is made up of again PET, which is hydrophilic coated. Okay, this PET is coated with hydrophilic coating, and so that it is hydrophilic, you understand, right? Hydrophilic is water loving. Okay, so if blood sample is kept, it is not, it will suck in the blood and it will give you the result. For that purpose, we use such kind of material. Okay, so these many layers are there in a glucose test strip. Okay. Construction and working. See how it look like. First figure we'll see, which is the simplest one to understand. Okay, here we have three electrode system. The center one will be the working electrode. Whatever explanation I gave, so many layers, that is for working electrode. Okay, that is the center part, and along with that we have a this curved part, which is a reference electrode, and the other side we have the counter electrode. And this is the complete test strip, and here if you put this strip into the display machine, it will give you the readings over there. Okay, so this is a three electrode system setup which is made. So this is a simple way to understand it. So what is it in detail if you want to know? So here the round portion in the right hand side figure, we'll move to right hand side figure. This round portion is where we actually apply the blood sample. Okay, and you can see this nylon mesh which I explained is there here. Okay, and we have the electrode which is made up of carbon indicator. Okay, for a current collector we want a conductor, right? So carbon, indicator electrode, one is used, the other side also, another electrode is also made up of carbon electrode, which is indicator electrode 2. Okay, and in between we have the working electrode where we have applied all the coatings, everything, and we have here 3 electrode assembly, and where we have the reference electrode also in between. Okay, reference electrode and the working electrode measures the potential, and working electrode and the counter electrode measures the current. That is the purpose of 3 electrode system. From the glucose value, see here, generally people doesn't know the range of glucose to be in the blood. Okay, when you are fasting in the morning, normally it should be in between 80 to 100. Okay, after eating, it comes to a high level of 170 to 200. Okay, after 2 to 3 hours, after digestion, after 2 hours, okay, it should be 120 to 140. That is the normal range. Okay, if you go for blood test, before fasting they take, and after eating, after 2 hours, they take the reading. Okay, then we have impaired glucose. Then we have, this is the range, fasting is 100 to 125, and after eating it is 230. And then we have, after 2 to 3 hours, we have 140 to 160. If you are diabetic, it is more than 126 on the fasting. And after eating, we have 20 to 300. And after 2 to 3 hours, it is 200 plus. Thank you, students. I hope you have understood the concept very well, how a glucose sensor works and how a blood sample is tested. Okay, let us meet on yet another topic, maybe which may be interesting for you. By now, thank you.